Hi everyone, welcome to this AP Environmental Science lecture on pollution and human health, but mostly focusing on pathogens and infectious diseases. So we got this one learning objective on pollution and human health, and the enduring understanding is that pollutants can have both direct and indirect impacts on the health of organisms, including humans. The learning objective is to identify sources of human health issues that are linked to pollution, and because it's really talking about sources of human health that are linked to pollution, like it says, we've already talked about this. Most of this stuff we've already talked about in different PowerPoints, whether that is PowerPoints talking about energy production or talking about different types of pollutants and their health impacts. We've really hit this in many different places. What we will talk about in this PowerPoint is dysentery. We've already talked about mesothelioma, so we're not really going to talk about that. We're not going to really talk about respiratory problems and lung function being impacted by high levels of ozone because we already talked about that. But we will just barely mention the difficulty to establish a cause and effect relationship between pollutants and human health. We've talked about that in class, how it's really difficult to do human studies. Like you cannot, for ethical reasons, expose people to varying levels of pollutants and track them in a controlled study. Okay, so for many reasons, it is difficult to determine a 100% direct relationship between the cause and effect of pollutants and human health issues. Where we're going to spend most of our time is talking about pathogens and infectious diseases. We're going to hit all of these learning objectives or essential knowledge. Here's the vocab if you want to pause it and jot it down. So like I said, you guys are responsible for knowing the different human health impacts of the various pollutants that we've talked about, whether those are atmospheric, aquatic, or terrestrial pollutants. So revisit those PowerPoints because, again, we've already talked about the human health impacts throughout this entire school year. Okay. For the second bullet point here, it is really difficult to know the negative health impacts of many of these pollutants. The chemical industry is creating new compounds all the time, and testing does not keep up with the creation of new compounds, and most testing is industry funded. So industry does have a clear conflict of interest when they create a new chemical and they find all of these beneficial properties of a new chemical. Those beneficial properties may be outweighed in their mind may outweigh in their mind the negative health impacts. Plus, they get a lot of money from marketing these chemicals for whatever uses that they are marketing them for. So there's a conflict of interest there, and they may hush up studies. They may pressure scientists to fudge results, to misinterpret data intentionally. And it's really difficult for the general scientific community to see their results because all of those are proprietary studies. Industry studies are proprietary and they own that data set. They do not have to publish it in peer-reviewed scientific journals like a scientist at a university would. Okay. The other issue that we have is acute versus chronic exposure. It's really difficult to nail down whether somebody is exposed to something like if somebody's exposed to something once and they get acute exposure to a toxin and they die from it or they get a some other negative health impact from it, that's pretty easy to tell. But if it's chronic exposure, low levels of a pollutant, parts per billion, parts per million, it's really difficult to tell if somebody gets this disease because they've been exposed to something for 30 years at very low levels. We also do most of our studies on mice and rats. We also do studies on primates, less often today, but we still do, to see if different chemicals are toxic. However, those proxy models may not represent human physiology 100%. They are very good models for human physiology because pretty much all biochemical processes in any mammal is the same, but it may not be 100% applicable to humans. 99.9, but maybe not 100. When we mix different compounds in the environment, we can have greater effects than if we were just to be exposed to a single compound. And most of these tests are done with just single compounds. But if we have a cocktail of compounds, let's say in drinking water, we might have more damage done to the human body than what we would expect from just adding up the sum of those parts from the sum of those different chemicals. All right. So with all that being said, regulatory bodies should be practicing the precautionary principle when it comes to new chemicals that are introduced into the consumer space. You guys can read the definition of the precautionary principle there, but as we have seen numerous times, the US does not do this. We essentially allow industry to develop a chemical, run its own industry-funded studies, and then without double checking those studies, put a new chemical into the marketplace where people can be exposed to it for years and in many cases decades, and in some cases many generations, several generations exposed to it, before we determine that there are negative effects through the relatively long process of scientific experimentation and peer review and repeating studies. The EU, on the other hand, at least in theory, this doesn't actually happen all the time in real life, but at least in theory, only introduces new chemicals once they've been proven safe. Whereas in the US, we have a safe until proven guilty type of thing, like innocent until proven guilty, safe until proven harmful, and we'll use it for years until we determine that it is harmful. And then we'll retract it, only to replace it with another chemical to replace that cycle. All right, so that's all we're going to talk about human health impacts for this PowerPoint. But again, revisit the old PowerPoints to see the negative health impacts of all the various pollutants that we've talked about. Let's start talking about pathogens. So a pathogen is any organism that can cause disease in another species. Those can include viruses, bacteria, fungi, protozoans. So those would be protists, such as malaria, and then some animals. These can occur in many environments, regardless of whether the environment looks clean or not. And let's talk about some of these pathogens. But before we do that, let's talk about the adaptability of pathogens. So all living things are going to have their ecological tolerances, and pathogens are no exception. They have their own ecological tolerances. Most pathogens are specific for a specific host, right? You're not going to have a parasitic wasp that preys on aphids all of a sudden start going after spiders. They are specific for specific host organisms. Although I might not consider a parasitic wasp a pathogen, but whatever. You will not give a tree a common cold. Okay, so that would be another example. Coronavirus cannot infect trees because they are mammal specific, but more importantly, they are human specific. There are some cases where pathogens can jump from one host to another, and we'll talk about those, so-called zoonotic diseases that originate in animals and then secondarily infect humans. But for the most for most of the time, pathogens are specific to a specific host or a specific small set of hosts. However, pathogens are notorious for being rapidly mutating. They are constantly in a evolutionary arms race with their host. Their host always wants to out-evolve the pathogens, and the pathogens are always trying to keep up and mutating rapidly to do so. Bacteria can perform horizontal gene transfer. So bacteria do not do sex, but they can, two bacterial cells can go next to each other and create kind of a little tunnel with their plasma membranes going through their cell walls and trade genes back and forth, especially genes that are on plasmids. So they'll move plasmids back and forth. So we can have a gene that causes resistance to penicillin in one bacteria being transferred to another bacteria, often of the same species, but not even of the same species in many cases. Viruses can also recombine within cells. So if two similar viruses infect the same cell, so let's say if a human lung cell gets infected with both a swine flu and a human influenza virus at the same time, those two virus genomes can get repackaged into one virus and then released when that cell ruptures to release those viruses. However, despite the mutability of pathogens, there are some pathogens that we have more or less eliminated. The greatest example of that, the best example of that is smallpox. There was a massive smallpox campaign in the mid-20th century to eradicate it around the world, and it's believed to be eradicated by 1979. And the only smallpox that exists today, as far as I know, is in labs. The other vaccine triumphs include polio, measles, rubella. Even though we have some instances of those, they're not completely eradicated. We have very, very, very low numbers of measles, mumps, rubella, etc., because of vaccination campaigns. That's not to say that every single campaign that we've done against pathogens have been successful. For example, the malarial campaigns of the 1950s and 60s were unquestionable failures. They were disasters, but malaria is a very different pathogen than these viruses were. All right, one thing with environmental science that is really important with connection to disease is the incidence of emergent diseases and how emergent diseases have increased throughout the 20th and 21st centuries. So an emergent disease is a disease that is new to the human population. They're usually jumping from animal hosts to secondarily infect humans. So they're an example of the zoonotic diseases that I mentioned before. And just a few highlights. You guys can see the list of the emergent diseases that we've had throughout the 20th and 21st centuries so far. Swine flu, the Great Influenza, the Spanish flu of 1918 and 1919, killing hundreds of thousands of people worldwide, coinciding with World War I, or at least the end of it. The virus that causes Ebola, first noted in 1976. Shortly after that, the virus that causes AIDS, so the HIV virus, we've been living with that for the past 43 years. SARS-CoV-2, definitely very familiar. You guys are very familiar with that. However, there is some debate about where that truly originated, but it is still an emergent disease, whether it originated in an animal or if it originated in a lab. But I want to highlight one of these emergent diseases, which is Bolivian hemorrhagic fever, or machupo. This emerged in the 1960s. So what I want you guys to imagine is that you're in South America, you're in Bolivia, you're on Bolivia's eastern frontier. So if we look at the map of Bolivia, we're seeing the Andes Mountains running through the more western portions of Bolivia, and then the north and eastern portions are getting down into the Amazon basin. And imagine that you're in a small town. It's basically like a cowboy town. It's a frontier town. There's dirt roads. There's no running water, no electricity. Imagine basically like, you know, early 1900s cowboy town in the United States. Okay. And in this town of San Joaquin, there is a fever that starts to break out, and people start to bleed from their nose, their mouth, their eyes, and everything, and then people start dying. And when the health authorities got there, they found that this was a new virus that was coming from mouse populations in that area. And essentially what they were doing as they were moving further and further and further with these frontier towns into what was previously natural habitat, previously untouched forest and savanna, the mice populations were moving into people's homes. You were destroying mouse habitat, and rather than the mice just dying, they moved into people's homes, and they had a ready food supply of corn for the most part in these people's homes. So they would they would get out at night, eat the corn, they would pee, they would defecate on the corn, on their sheets, on their floors, on their wherever, and this virus was transmitted through their urine to people. So they were even the dust from the urine, like the mouse pees on the floor, you sweep the floor and you inhale this dust, you can get the virus transmitted to you in that way. So as people are moving further and further into natural habitats, we are becoming more and more exposed to populations of different organisms, and we're increasing the incidence of, or at least the possibility of, viruses jumping from one host to another, in this case from a mouse to a human. Now this virus is a hemorrhagic fever, meaning that the capillaries essentially fail. I believe in this case, the diameter of the capillaries increases so much that the lining of the capillaries becomes really, really, really thin and allows blood to leak into the interstitial tissue. This leads to headache, fever, fatigue, bloody stool, bloody vomit, blood in your airways, so you're coughing up blood, and people start to bleed from the nose, the mouth, the eyes, and you have excruciating pain all over the body. And about 50% of the town of San Joaquin was affected, and between 25 and 50% of the infected dies, so upward estimates of 25% of the town dying from this. Um, it is more or less controlled today. We know where it comes from. We know what actions to take to keep the mice out of the homes. But this is what I'm talking about with emergent diseases, a disease that was never in the human population before, or at least never documented in the human population. And then as we're moving further and further into natural habitat, we are becoming more and more exposed to these animal populations so that diseases can jump. All right, but Machupo Bolivian hemorrhagic fever is not one that you need to know for the AP test. These are the ones that you need to know for the AP test. So this is what's on their AP standards, and these are the ones that we're going to focus on. So just getting right into the first one, plague. This is known as bubonic plague, septicemic plague, the Black Death. You've probably heard about this in history class. This is caused by bacteria that resides in fleas. The bacteria is Yersinia pestis, and the vector is the fleas. So a vector is is like an animal that can transmit the disease, not specifically the animal itself, but the animal is carrying the disease, the pathogen, the disease organism, and can move it to humans. So the vector in this case is fleas. Rats or any other rodent carried the fleas, so people thought that rats got people sick, but it was actually the fleas on the rats that would jump from the rat to the person and then bite the person and give them the plague bacteria, infect them with the plague bacteria. Now, this is environmentally dependent because the fleas can pass the bacteria at high temperature, meaning that they get rid of the bacteria in their feces, but the bacteria are going to lodge in their gut at low temperatures. It's going to make them feel like they're constantly hungry. The fleas are going to be constantly hungry, and they're going to bite more and more and more people. And because that bacteria is in their gut, they're going to give people that they're going to inject people with that bacteria, or the bacteria is going to infect the person's wound around the mouth parts of the flea, and they're going to infect the person with the bacteria and cause plague. All right, and we're going to skip most of the history stuff, even though it is really interesting. I have it up here if you want to read it. But plague is currently endemic to North and East Africa. It's endemic to Eurasia. It's endemic to North America. So plague is pretty widespread. And again, it's still carried on fleas on rodents, including prairie dogs here in Colorado. So I would never approach a prairie dog because if you get fleas on them, from them on you, it could carry plague. Not a guarantee, but it could. Okay, the plague bacteria, the Yersinia pestis bacteria, is going to proliferate. It's going to reproduce in your lymph nodes, and that's one of the most insidious things about this bacteria because your lymph nodes are part of the lymph system, the lymphatic system that is responsible for killing and removing the pathogens from the body. So they proliferate in the same organ that's responsible for that. They're basically immune to many of the white blood cells, including macrophages, which are responsible for phagocytosing pathogens. They basically engulf or eat pathogens. You probably remember that term from biology class. So macrophages will engulf or take in foreign bodies, and in this case, a bacterial cell. Okay, but the Yersinia pestis bacteria is, let's just say, immune to that. The symptoms of plague, you can, in increasing order, first get bubonic plague. This is where you get fever, headache, chills, swollen and painful lymph nodes, and those swollen lymph nodes are called buboes, which is where bubonic plague comes from. The second part is septicemic plague. This is when the bubonic plague goes further and gets worse. And that's going to be fever, chills. The skin is going to turn black and die. So you see an example of somebody's hand up in the top that has septicemic plague. And you can have internal hemorrhaging, and it can be fatal. And then finally is pneumonic plague, and that's where the bacteria is going to get into the lungs, and it's going to cause fever and pneumonia, and the mortality rate is above 90%. So it's essentially a death sentence to get pneumonic plague. And it's at that point that it can be transmitted from one person to another via respiratory droplets. So that's a secondary way that plague can be transmitted, is through respiratory droplets if it is infecting the lungs as pneumonic plague. right, speaking of the lungs, let's talk about tuberculosis or TB. This is also known as consumption. So if you read a old book, especially written in the early 1900s or before 1800s, 1700s, they'll often call it consumption. Even Shakespeare, I believe, called it consumption. This is caused by the bacteria Mycobacterium tuberculosis. So it is a bacterial infection. You guys can see the symptoms up at the top right for people that have active TB. This is going to spread through respiratory droplets. So you guys are very familiar with those from COVID, but it's coughing, sneezing, even talking and spitting whenever you have micro droplets of saliva that are getting ejected from the mouth through any of these activities. It's estimated that about a quarter of the world population has tuberculosis, but it's latent tuberculosis, meaning that they are infected, but the immune system keeps that infection under control and they show no symptoms whatsoever. So the bacteria is in the lungs, but the immune system is preventing any symptoms from being shown. It's keeping that bacteria in check. But if somebody becomes stressed, if their immune system gets stressed in any way, or if the bacteria proliferates so much that it overrides the immune system, they can develop active TB. And this is where you have heavy chronic coughing, you often cough up blood, you have fever, and you have weight loss. And that's also why it's called sometimes wasting disease. And that's where consumption comes in, because it's like the body is wasting away. About 10% of latent TB cases become active, and active TB cases have a 50% mortality rate. Now, where does this play into environmental science? Primarily with them being drug-resistant bacteria. So TB is notorious for developing drug resistance. So because it's a bacteria, it is often treated with antibiotics. However, those antibiotics can lead to the natural selection of drug-resistant tuberculosis strains. And once one strain becomes resistant to one drug, you might use a second drug, which would lead to multiple resistant tuberculosis. And then you also have extensively drug-resistant tuberculosis or totally drug-resistant tuberculosis. We have seen strains of any of these from the mid-1980s, but they're becoming more widespread and common throughout the 2000s. You guys can also see where the incidence of tuberculosis around the world is. We've mostly eliminated it through the United States and the rest of North America. However, it is still very prevalent in Africa and Asia. All right, so how do bacteria become antibiotic resistant? It's basically the same process that we talked about pests becoming pesticide resistant, whether that is weeds becoming herbicide resistant, like we talked about with glyphosate, or if it's resistance to any other pesticide. So you start off with a population of bacteria in this case. And in those bacteria, maybe one of them just mutates to become randomly antibiotic resistant. It's a random mutation, but then we introduce a selective pressure. That selective pressure is the antibiotics that kill all of the other bacteria, and that one that is antibiotic resistant survives. When it survives, it replicates, it multiplies, and you form a population of antibiotic-resistant bacteria. And those bacteria can perform conjugation and do horizontal gene transfer and pass those genes from one bacterium to another. Sometimes, most of the time, the same bacterial species, but oftentimes it can be different bacterial species as well. This can happen to develop drug-resistant tuberculosis, multiple drug-resistant tuberculosis, etc. It can also be occurring in different populations of bacteria, whether those are E. coli or any other bacteria. We can have antibiotic resistance, but in the context of tuberculosis, antibiotic resistance is what you should know. So the next disease that we'll talk about is malaria. Malaria is caused by several species of Plasmodium parasites. These are not prokaryotes. They are not bacteria. They are instead eukaryotes. They are in that kind of fifth kingdom of life that we use as a junk drawer for basically anything that is single-celled eukaryotic that isn't clearly a plant, animal, or fungus. So it is a protozoan. Okay. You don't need to know the species that are involved, and you don't really even need to know that it's Anopheles mosquitoes, but the vector for the malarial parasite is female mosquitoes. They will bite you, inject you with the malarial parasite that is contaminating their mouth parts. Okay. Passing it from one person to the next via their gut. It's estimated that about 400,000 deaths occur annually from malaria. Most of those are children, about 2/3 of all those deaths. And the fatality rates vary by age and previous exposure. If you've been exposed before and survived that initial infection, then you're going to be more likely to recover from any subsequent infections. Plus, there is different species of the Plasmodium parasite with different mortality rates, which we don't really need to go over. The Anopheles mosquito that transmit malaria is a tropical mosquito. So you notice that malaria is confined to the tropics. There used to be cases of malaria in the southern United States. So if you read old literature like from the 1800s, you'll see that malaria cases occurred in the Gulf states, Florida, and in the southern United States. However, today we don't have many malaria cases in the United States at all. It is really confined to the tropics. However, that is expected to change. That malaria is expected to start having higher instances in southern Europe, Australia, southern United States as global warming occurs and the range of these tropical and subtropical mosquitoes extends into higher latitudes. So that's really what the environmental impact of this is, or the environmental connection, is that malaria and other tropical mosquito-borne diseases are expected to move into the higher latitudes as the planet warms and those mosquitoes' ranges expand into those higher latitudes. So there's a number of efforts that we can take to prevent the spread of malaria. There are a number of drugs that are used to treat it first off, but there are drug-resistant populations of those plasmodium viruses that have existed for decades at this point. There is no vaccine for malaria, and vaccination efforts have largely failed or totally failed. There are preventative measures though. The most common preventative measure is mosquito nets and other type of screens, so like screen doors, screens on windows, those types of things. You can use insect repellents, which can come with their own unintended consequences. You can use insecticides, which definitely come with unintended consequences, especially the elimination of non-target species, and stuff like DDT still being used to spray for mosquitoes, despite its well-known and well-documented environmental problems. Draining of standing water where Anopheles mosquitoes breed is another huge way to prevent the spread of the malarial parasite, because if you don't allow for those mosquitoes to breed in that area, then they're not going to be there to even transmit the disease in the first place. However, as we see from this quote by Dr. Win Kalami from the Director General of Tanzania's National Institute for Medical Research, saying that what can you do when these mosquitoes can breed thousands of offspring and they puddle the size of a hippo's foot? During the rainy season, there's absolutely nothing, and then you see the scale of a hippo's footprint versus a human's footprint or a human's foot, and you can have thousands of mosquito larvae in just that one small pool. So during the rainy season, it's really impractical to drain all standing water because it's too big of an effort. It's impractical. However, there are some ways that we can prevent malaria in terms of draining water, such as tires. So used tires that are dumped should be slashed before dumping so that they will just drain water. You should not have rain collection barrels or buckets or any type of standing water in malarial zones when they are in the breeding season. The mosquitoes' breeding season is basically all year round. But if you do have those, they should be covered. They should be inaccessible to the mosquitoes. Another thing that is being used that is not up here is releasing sterile male mosquitoes. And as sterile male mosquitoes will breed with females, they won't produce any offspring. They won't produce any eggs that are fertile. And that will reduce population numbers. So you can genetically modify male mosquitoes to be sterile, release them in large numbers so that they're a decent or significant portion of the population, and thus the females mate with them and lay sterile eggs or don't lay any eggs at all. Our next disease is West Nile virus. This is the same viral genus as several of our other tropical and subtropical diseases that you see listed here. This is a zoonotic disease, so the rest of the ones that we've talked about have been old diseases that are endemic to humans, but this is our first zoonotic disease. This is a disease that originated in animals and then jumps to humans. So humans are not the normal hosts. Humans are a secondary host. The event of when a zoonotic disease jumps from a primary animal host to humans is called spillover. So that's the vocab word for that. The normal host are over 300 species of birds, and the vectors are mosquitoes, but in this case, the genus Culex mosquitoes. West Nile virus can infect humans, other primates, dogs, cats, horses, but most of those are dead-end hosts, but they can have mortality rates. In humans, about 80% of the infected will show symptoms. 20%, will show no symptoms. 80% of the West Nile cases in humans will show no symptoms, and most people won't even know that they have West Nile virus. 20% will develop West Nile fever, and less than 1% will develop severe symptoms. So it's small portions that develop severe symptoms. We can contrast that with horses, where over 40% of US horses that have been infected have died. So it really is species specific about what the mortality rates are. West Nile virus was first identified in Uganda in 1937, which is why it's called West Nile, because it's identified from around the White Nile, which is the West Nile, so named after the river. There was a rapid spread out of sub-Saharan Africa in the 1990s. It was detected in Morocco, so jumping across the Sahara Desert by 1996, and then in Southern Europe and Israel, the Middle East by 1998, and the US in 1999. Now it is endemic to large portions of North America, including all the three largest countries in North America, and much of the Middle East and Central Asia, Australia, some South Pacific areas as well. Now, what do I mean by a dead-end host? So a dead-end host means that once a human is infected with West Nile virus, if another mosquito bites that human and sucks their blood out, they will not get the West Nile virus, and they will not be able to transmit that West Nile virus to another person. So from birds, you can go from bird, mosquito, back to bird. Once it gets to humans, you can't go from human to any other organism. Okay, so it's a dead-end host. The next couple diseases that we'll talk about are SARS and MERS. SARS stands for Severe Acute Respiratory Syndrome. This is caused by a virus called SARS-CoV-1. You might recognize that. This is a zoonotic disease that originated in horseshoe bats in Asia and then went to a Asian palm civet and then to humans. So there was that intermediate host between the primary host of the bat and then humans. This was first identified in China in 2002. The outbreak was in 2004. And there were no cases since 2004, so it's believed to be eradicated in humans. However, we are monitoring the situation worldwide and keeping sure that it is doesn't occur in humans again. Between 2002 and 2004, there were 8,422 people that were infected, and the case fatality rate, meaning that of the people that were infected dying, it was 11%. This is the reason that we were so worried about SARS-CoV-2 or COVID-19 when it emerged, because even though the case fatality rate was much, much lower, I've seen estimates anywhere between 0.5 to 3.5% for SARS-CoV-2. We, at the very beginning, took a lot of precautions because what if the case fatality rate was 11%? What if 1 out of 10 people that's contracted COVID-19 ended up dying? That would have been much, much, much worse of a pandemic than it was. So that's why we took so many precautions at the beginning, because this was the model, the only model that we were able to use as a baseline comparison of like, what is this new virus COVID-19 going to turn into? So this spreads just like COVID-19 through respiratory droplets. The symptoms will include fever, cough, muscle pain, complications, and leading to secondary infections such as pneumonia. Pneumonia is actually the biggest killer here because pneumonia is a secondary infection and leads to, in many cases, just more complicated and more severe respiratory issues that can lead to death. And then MERS is a very similar type of coronavirus. It is the Middle East Respiratory Syndrome, and it's the virus itself is the Middle East Respiratory Syndrome related coronavirus, which is definitely a mouthload. And this was first identified in 2012. There were over 2,500 cases worldwide, mostly in the Arabian Peninsula, and this had case fatality rate of 35%. So imagine if 1 out of 3 people that contracted COVID-19 died from it. So again, SARS and MERS is why we took COVID-19 so seriously until we were able to ease lockdowns and ease restrictions, because we determined that the case fatality rate was much, much less than these other two more transmissible viruses. Or sorry, these other two much less transmissible viruses. SARS-CoV-2 is super easily transmitted, MERS less so. So it has never really escaped the Middle East that we know of. All right, another emergent disease is Zika virus. The vector for this is Aedes genus of mosquitoes. Again, you don't need to know the genus of mosquitoes for this class, but I have it up here anyway. And this can be sexually transmitted, so it is a sexually transmitted disease as well. The first instance of Zika virus was identified in 1947 in the Zika forest of Uganda, which has 2 eyes, whereas the virus just has 1. I don't know why. And it's known from equatorial Africa and Asia since the 1950s, and then spreading to the Americas in 2007, most likely due to either humans that contracted it moving across, or mosquitoes that were were stowaways on ships moving across. The symptoms, the adults, they're typically none, but you can have a mild rash, fever, joint pain, etc. And it can, in very rare cases, lead to Guillain-Barré syndrome, which is the rapid onset of muscular wasting. But the main concern for Zika is with pregnant mothers. You have vertical transmission from mother to child during pregnancy, and this leads to microcephaly in babies. So you see a baby with microcephaly here, and then a baby with severe microcephaly, and the dotted line showing how big the brain should be. Obviously, if you're born with a much smaller brain than usual, you're going to have very severe cognitive impairment. As with other tropical mosquito-borne diseases, the reign of Zika is expected to expand into higher latitudes with climate change. So just like we talked about with malaria. The last couple illnesses that we'll talk about are both gastrointestinal or diarrheal illnesses. The first is cholera, which is caused by the bacteria Vibrio cholerae. This is transmitted via contaminated water that is contaminated with human fecal matter. It can also less commonly come from eating undercooked crustaceans like shrimp or crab, because these bacteria can attach themselves to the crustacean's chitin exoskeleton and then be transmitted to humans that way. It can also form on spore-like forms on the inside of algae, like inside of algal cells, inside of cyanobacterial cells, and be transmitted that way as well, and eventually get into a human host. However, that is much less common. So the 2 ways are coming from water that is contaminated with human fecal matter, and then from seafood, specifically crustaceans, where that bacteria can attach to their chitin exoskeletons and then be ingested if it's raw or undercooked. This is a very old disease that is endemic to the Indian subcontinent and Southeast Asia. There are no known animal hosts of the disease. It's just a human disease. The symptoms include violent watery diarrhea, and that's how most people die, is through the dehydration that is caused by the diarrhea, as well as the electrolyte imbalance. So you lose so much fluids that you end up dehydrated and die. The case fatality rate among severe cases is between 25 and 50%, depending on treatment. There's an estimated 3 to 5 million people affected annually, with 25,000 to 135,000 deaths. But again, on severe cases, 25 to 50% mortality rate. The treatment is just going to involve waiting it out and giving the patient lots and lots of fluid. So when we talk about cholera and the next one, which is dysentery, clean drinking water is really the main solution for this. So proper sanitization and clean drinking water, whether that is coming from a water treatment facility and then pumped to people's homes, or whether it's coming from boiling your water, or if it's coming from individual water filtration systems. Okay, having proper sewage treatment is also critical, and getting proper sewage treatment and clean municipal water supplies to people in developing nations is really the best way to prevent and to prevent cholera from occurring. You can also have, like I said, individual water filtration systems. So you see two examples of those up here on the top. The first is a device called a life straw, which you actually may have if you're an avid camper or backpacker. I have one for when I'm camping, and it's definitely been used, like paid for itself in use. Basically, what you can do with this is that it has several layers of filters are the pore sizes are smaller than the bacteria, so it will capture that bacteria through the filtration system in this as you're sucking water through this life straw from whatever dirty source it is. Another example is shown here on the kind of the middle, where we have a filtration system that is doing the same thing but on a larger scale with very small micron filters and making clean water down at the bottom. Okay. There has been, you guys don't need to know the history behind cholera, but there has been 7 major cholera pandemics in the last 200 years. So when we're talking about a pandemic, we're talking about a global epidemic. So there's been many smaller epidemics as well, but just in the last 200 years, there's been 7 major cholera pandemics. So the last disease that we'll talk about is dysentery. Dysentery is not a single disease, but rather it's a group of diseases that all have similar symptoms, which include inflammation of the gastrointestinal tract, which leads to bloody diarrhea. And most people are going to die from dehydration, just like with cholera. It's a very old disease, and again, it's caused by several types of pathogens. You don't need to know any of the specific pathogens, but it is caused by many pathogens such as Shigella, Giardia, which you might have encountered in Colorado. I know I've gotten dysentery from Giardia before, camping and drinking water that I thought was clean, and it wasn't clean. And I didn't have a life straw at the time. The life straw would have prevented that, but I was drinking fresh snow melt in a creek. Being stupid. Anyway, the sources of the organisms, contaminated water, are going to be exactly the same for cholera, except this is a more worldwide and it's a little bit more prevalent because there's more organisms that can cause dysentery than cholera. The solutions for it are the exact same as well: sanitation, clean drinking water. Okay, and you can see the number of cases and the deaths every year. Most of those are going to be in the developing world because that's where wastewater treatment facilities are and water treatment facilities are less common. All right, so why did we go through all of these diseases? Why are we talking about these in an environmental science class? Well, they have some common environmental themes. These are in no specific order, but I just have them up here in this order. The first one is sanitation and clean drinking water are critical to controlling waterborne illnesses. Dysentery, cholera, you need to have proper sanitation and clean drinking water in order to control for those diseases. Wastewater treatment facilities, drinking water treatment facilities. Until we get those in every community worldwide, we are going to have incidences of cholera and dysentery. Number 2, as the climate changes, the ranges of pathogens and their vectors is going to shift. You're going to have tropical and subtropical illnesses expanding into the higher latitudes as the tropical and subtropical mosquitoes can survive in those higher latitudes as the planet warms. Okay. For number 3, pathogens evolve rapidly and some can become antibiotic resistant and even vaccine resistant. The overuse of antibiotics in our medical system has accelerated this. Okay, so just like we talked about with tuberculosis. Number 4, increased penetration into wild areas exposes humans to zoonotic diseases, which have the potential to jump to humans. So as we destroy habitat and as we encroach further into wild spaces, we have the potential to encounter more and different types of animals for them to invade our homes, although I would argue that we are invading their habitats, and for those diseases that are for millions of years been in just this one animal host to potentially spill over to the human population. Number 5, global travel rapidly spreads pathogens. So we saw this with SARS-CoV-2. Global travel is going to easily spread pathogens all around the world. So even though we didn't talk about it in the lecture, it's here. The world is becoming a smaller place, and you can go from one continent to another in the matter of hours to days, and you can bring your pathogens with you. And then number 6, high population densities, including those that come with urbanization, lead to rapid spread of disease. And we talked about that mostly in the context of animals and agriculture, but whenever you see high population densities, whether that is humans or animals or plants, know that disease can spread really, really quickly as opposed to when you have low population densities. All right, for the learning objectives, again, we talked about several of these in different lectures, so I'll have you go to those different lectures to see some of them, but we did talk about dysentery, and we did talk about some of those ways that it's difficult to establish a cause and effect relationship between pollutants and human health issues. And then finally, the pathogens and infectious diseases. So pathogens adapt to take advantage of new opportunities and infect and spread through human populations. Hopefully you saw a little bit of that in every single one of these, or at least most of them. Specific pathogens occur in many environments, whether or not that appears clean. And then we talked about how as climate change shifts, the range of those disease vectors is going to shift as well. And then low-income and poverty-stricken areas are more likely to have dysentery, cholera, or any other waterborne illness. We talked about plague, we talked about TB, we talked about malaria, we talked about West Nile, SARS and MERS, Zika, and cholera. So if you guys have any questions, please let me know. Hope you learned something new, and I'll see you all in class. Bye.